Alright guys, welcome to our digital learning platform. Today we're going to talk about protein synthesis. But before we can talk about protein synthesis, we have to talk about DNA and RNA. Now, DNA we've already talked about, and we know that it has 4 bases, A, adenine, T, thymine, G, guanine, and C, cytosine. We also know that DNA is a double helix, so it's got 2 strands that come together to form our double helix. Now we have RNA. And RNA is a lot like DNA. It has A's, G's, and C's, but it's different because instead of thymine, it has something called uracil, or U. It's also single-stranded. So now that we've reviewed DNA and RNA, let's talk about protein synthesis. Protein synthesis has 2 steps, transcription and translation. Transcription is when we move from our DNA molecule and we convert to an RNA molecule. And so we start, if you remember when we did it, we split our double helix into 2 pieces, and that's going to be copied by an RNA strand. Now, once that goes, it moves from the nucleus out to the ribosome, and it goes through translation. And what happens in translation is it reads the RNA. And it does that by separating the RNA into its individual nitrogen bases and cutting them in to 3s. And one 3-letter section is called a codon. Now that we know the general of the DNA goes through transcription to become an RNA molecule, RNA then leaves the nucleus and heads to a ribosome to then go through translation to become an amino acid chain, which are the building blocks of my proteins. Let's take a more detailed look at what happens as we move between those steps. So, DNA starts with our molecules C, A, T, and G. We then need to convert those to RNA, so my C's become G's. But remember, T's, thymine, were replaced with U, uracil, when we go to RNA. So A's become U's, T's become A's, G's become C's, C's become G's, A's become U's, T's become A's, C's become G's. I then need to convert my RNA into amino acids. And the way I do that is I break them into codons. And those codons are always 3 long. Each codon has 3 pieces within it. We have a first position, a second position, and a third position, and we're going to use those positions to figure out what each codon codes for. So, my first position of my first amino acid codon is G. So I'm going to circle G. My second position right here is U. So I'm going to circle the U column because this is labeled second position. I then take a look at my third and I find it where they overlap. And if I see, that leaves me with the amino acid VAL. Still confused? Let's try our second set of 3. We have C, G, U. And so we're going to be looking for our second amino acid. This side, right here, is labeled first position. And my first position is a C. So I'm going to circle that entire row. My second position, and it's labeled second position here, is a G. So I'm going to circle that entire column. And I'm going to find where those 2 columns and rows overlap. I then look at my last letter, U, to determine that my amino acid is ARG. I want everyone to pause the video now, and I want you to try and figure out what our last amino acid would be in this chain. Were you able to figure it out? If not, let's take a look. We have 3 letters, U, A, G. Our first position is a U, so I'm going to circle the row U. My second position is an A, so I'm going to circle the column A. And my last position is a G, so I look where they overlap and I find G. And my codon is coding for the word stop. And what stop means is that it's done making my amino acid chain, and I have created a protein. If you have any other questions, guys, I'm here for office hours between 1 and 3, Mondays, Wednesdays, Thursdays, and Friday. Don't forget to do your work, posted both Wednesday and Thursday, and take your 10-question quiz on Friday. All of this is due on Saturday at 11:59. I hope to hear from you all soon.